Science for Malaria Impact webinar series. We welcome you to the fourth session, a platform to amplify the voice of leading researchers in malaria endemic countries. And what our hope is, and we've been very pleased with the uptake, we aim to bring the malaria community of innovators and showcase the work of malaria endemic researchers. These are 1-hour sessions held monthly and cover a range of topics of relevance to the current challenges being faced in the field and the potential for science to address them. This webinar is entitled How to Tackle the Challenge of Insecticide and Drug Resistance with Innovative Approaches. And our moderator is Professor Abdoulaye Djimdé, who is the Kames Professor of Parasitology and Mycology at the University of Sciences, Techniques and Technology at Bamako in Mali. It has been my pleasure to get to know Dr. Djimdé since the MESA started, really. But he received a pharmacy doctoral degree from the École Nationale de Médecine et Pharmacie of Bamako, Mali in 1988. He spent a fellowship at the U.S. National Institute of Health. He received his PhD in microbiology and immunology, University of Maryland, and where he began really what has been a career-long exploration and seminal studies describing the first reliable molecular marker for chloroquine resistance, and he proposed a model for field monitoring of drug resistance using molecular markers. Since his return to Mali, he started his own laboratory, the Molecular Epidemiology and Drug Resistance Unit at the University in Bamako. And his goal of his research is to understand how the malaria parasite becomes resistant to malaria drugs and how that resistance spreads over time and space. Parenthetically, he's also played important leadership roles in the global malaria community, including being a member of the board of ASTMH, participating and having a very strong voice in the malaria refresh papers. So we are delighted to ask him and to have him join us to chair this session. I will now turn it over to Dr. Djimdé.
Okay, thank you, Regina, for this nice introduction. Good morning, good afternoon, ladies and gentlemen. Welcome to this webinar. It is my pleasure to moderate this webinar, and starting with introducing our first speaker. We'll be hopefully covering the malaria parasite and the mosquito vector today, and addressing issues of major importance in terms of resistance, the evolution, the spread, which are really important topics as we are moving towards elimination of this disease. The first speaker will be Professor Alfred Amambua-Ngwa, who Alfred I've known for a long time as a colleague, a friend, a fellow member of a number of networks, the Plasmodium and Pathogens Genomic Diversity Network, the DELGEM network, PAMGen network. Alfred and I really have been collaborating for a very long time. He's an associate professor at the Medical Research Council Unit at the Gambia at London School of Hygiene and Tropical Medicine. Alfred studied biochemistry and molecular parasitology at the University of Buea in Cameroon, earning a PhD degree in 2004. He spent 5 years of his early postdoc at the MRC unit of Gambia at the London School of Hygiene and Tropical Medicine, researching P. falciparum invasion pathways, genetic diversity, and adaptation to drugs and host immunity. He continues to investigate malaria populations using genomics approaches to interrogate Plasmodium, not only the falciparum, but the other species, ovale, specifically malaria, and a bit of vivax. Human and vector adaptation and malaria transmission across population. He is a member of several regional research development networks and a number of research projects, including the African Academy of Science funded PAMGen East Africa Consortium for Malaria Genetic Epidemiology, spanning 7 countries in Africa. Alfred is a true, I used to call him one of the rising stars in the next generation of African scientists. He's not a rising star anymore. He's a bona fide star in his own rights. I will be delighted to listen to Alfred today. You have the floor.
Thanks very much, Djimdé, for that elaborate introduction. I think there's no need for me to introduce myself again. No. I used to tell Djimdé that I remain a rising star because we still have Djimdé and many others like Professor Ogo of blessed memory that we want to emulate, want to follow their example. So Djimdé, please classify me still as a rising star. If I am a star, I'll be happy to take that. As Djimdé had said, I was trained at the University of Buea. The University of Buea is at the foot of a mountain in Cameroon. This is the Mount Cameroon, and this is the university. I spent 13 years training there, and I left in 2006. And ever since, I've been at MRC the Gambia working on malaria. So I'm delighted to present to you today some of the work we are doing. This is not work that is done by me. There's including Djimdé and contributions from many partners across the globe. Though I've made the topic malaria parasite populations, their transmission and adaptation across African endemic populations, there is much more that we can do for each of those sub-words that I've actually put in those titles. To this forum, I guess there is no need to preach to the converted. We know what has been happening to malaria over the last 2 decades with a report of significant decline. And this significant decline in the last 20 years has been due to the significant investment in interventions, especially insecticides against the vector in bed nets and IRS, as well as new drugs such as the artemisinin combination drugs that are being used, and other interventions with drugs such as the SMC. However, there are worrying signs that malaria decline might be stalling, and there has been a decrease in the rate at which the malaria parasite incidence has been going down across the continent. And some of the factors that contribute to this beyond the operational implementation and funding factors might actually be things that are related to the parasite biology or to the biology of the mosquitoes that are the vectors of the malaria parasites. Most importantly, drug and insecticide resistance. So one of the things we really want to do is to understand how parasites in populations, just like Djimdé, who I collaborate with, is interested in, is find out how parasites in natural populations do respond to drugs, how they develop tolerance. And the reason that we think this is important is because since the inception of ACTs, there have been several ACTs that have actually that are being used across the continent. And different populations are using different types of ACTs. So these put together, there is definitely a complex dynamics of interaction between the parasite population and drugs, just like the case for mosquitoes. And so it's really important for us to continue to look at this partially and also to look at this over time. Unfortunately, there are several methods that are being used to monitor the effect of the drugs on the parasites. But unfortunately, the data that represent this across the continent is very scanty. And you can look at this from the recently published paper by Hannah that is looking at molecular markers that have been surveyed across the continent. And what we see here is that there are very scanty positions across the continent where data is collected. And the reason for this is clear. There's lack of manpower to do this kind of analysis. There is also lack of the analytical skills to do it. And these are some of the things that need to be addressed. We need to improve on our capacity to continue to do surveillance at the molecular and the phenotypic level across the continent. Despite this small amount of data, what is clear from this publication is that there has been changes over the last decade, where we're seeing increased frequencies of wild-type mutations in some of the genes that are associated with drug resistance, resistance to previous drugs. And these mutations are allowing some of the parasites to be more tolerant to the partner drugs that are used in the current ACTs, such as lumefantrine. Now, these changes are influenced by several other factors, and some of it is the type of drug that's being used, the rainfall patterns, seasonality, for example, and the intensity of transmission that might actually determine the rate of recombination in the populations of parasites. So in order to actually move forward and increase the population of data, the density of data across the continent for the parasite as well as for the vector, the PDNA was formed by Professor Djimdé, who is the moderator of this group. And this network has been very instrumental in not only putting together the scientists that are interested in answering questions about the evolution of drug resistance and insecticide resistance, but also to bring together people with shared ideas and think about other things about the biology of the parasite and the mosquitoes that might be important for the malaria elimination programs across the continent. Several countries are represented in this network. This network has published 3 important papers in which I participated in some of the analyses. The first one was the one that described CLUS13 mutations in African populations led by Ed Kamau. And we showed that there were no mutations that are similar to the mutations in Southeast Asia that cause drug resistance or cause delayed clearance to artemisinin in our African populations. Now, the second publication that we had was also an important publication where we were describing the substructure of the parasite across the continent. And recently, we published one article that described the diversity of vaccine candidates that are being developed for future vaccines against malaria. So it's an important group that will continue to work on malaria, and there are several new articles that will be coming out very soon. Now, I just want to elaborate a bit on the publication that described the substructure of African parasites across the continent, which was led by me. Now, we took advantage of the fact that sequences have been made available to us by the Malaria Gene Program, thanks to our contribution to their objectives. And in this publication, we actually analyzed over 2,300 genomes of the parasite across 27 locations in 15 countries across the continent. What we showed, obviously, is that there are differences in the complexity of infection. You can see that by looking at the inbreeding coefficient or just by measuring or modeling the number of possible genotypes within a single infection. There is a huge variance across the continent. And some of these will affect the ways that the parasite responds to interventions. So what we showed using the data that we analyzed across the continent is that we do have subpopulations of the parasites that represent the West, the Central, and a very unique population in Congo that is quite admixed, a highly divergent population in Ethiopia that looks quite different from the rest of the continent, and those in Eastern Africa. Surprisingly, the populations from Malawi and Madagascar looked like a population of their own, irrespective of their proximity to Congo and to Tanzania. That said, the populations that we have described are equally admixed, in which case they're sharing haplotypes from each other. So parts of the gene flow from east to west and from west to east. And this is also important for drug resistance. As we know, drug resistance has been traditionally said to flow from the east to the west. That is true, it's largely true, but there are also other things that we're now finding out that may have emerged locally, like we are seeing in the case for artemisinin in Rwanda. So we are refining these analyses of population structure across the continent. And instead of using SNPs, we are now using microsatellites. And we are continue to break the populations into subgroups that might be important, that might be showing important backbones on which some of these drug mutations might have introgressed or might emerge. And so now using microsatellite, we're seeing not just 6, we're seeing up to 8 different groups. We're improving on this and seeing subpopulations in Eastern Africa, like the case of Malawi, where we're now seeing 2 groups, a group that is very similar to Madagascar and one that is unique to Malawi and Tanzania. We continue to see the Congolese population as an admixed population, or probably an ancestral populations where all the other populations are having ancestral haplotypes in that group. Now, why is this kind of analysis important to drug resistance? Now, we've tried to test this by just looking at the drug resistance loci or the SNPs or polymorphisms across drug resistance loci in the genome. And applying that similar analysis of ancestry to those SNPs, what we see is that the population in West Africa has 3 subpopulations based on their backbone of drug resistance around the drug resistant genes. And one of those subgroups is the one that is shared across the continent with all the other populations, in which case there are haplotypes of drug resistance that are shared across the continent, but there are also those that are unique to some populations. And even within the population of West Africa, we are seeing 2 divergent backbones across of drug resistance. And so it's important for us to continue to investigate this and determine how this might be affecting the responses to drugs, or putting at risk the possibility of putting at risk the current drugs with the possibility of developing drug resistance. So across the continent, when you look at across populations, you will see both strong differences at different haplotypes that are around drug-resistant genes, for example, the PSRA. But beyond this, you also see a lot of sharing of haplotypes across the same regions. So there is both unique haplotypes as well as shared haplotypes. So this is for CRT, that's for DHPS, that's responsible for SP resistance, CRT for chloroquine resistance. And we are now seeing currently emerging in Ghana and Malawi, for example, a sharing of haplotypes around the AP2 mu gene, a locus that has now been shown to have mutations that increase the ring survival of Plasmodium in vitro. So this is really important. So from genetic studies, we are showing that things that have been done in vitro are now being seen at the epidemiological level. We're also seeing shared haplotypes at genes that we know have not been clearly characterized as responsible for drug resistance. And one of those genes, the amino acid transporter on chromosome 6. Now, recently, we've been doing analysis on that particular locus in the Gambia. And what we have found is that if you look over time, for the last, since 1985 to now, it would seem that the mutations in PFA81, which is an amino acid transporter on chromosome 6, and those on chromosome 7 at the PFCRT locus are strongly linked. They seem to have emerged at the same time, and their evolution seem to be parallel, where there is positive selection, and now we are having almost a fixation of the mutant types in the Gambian populations. Looking across Africa for the PF80 locus, for example, we are seeing that the mutant variant is quite common across the continent. Except for a population like Malawi, there has been a reversal of the PFCRT to the wild type. And we are also seeing here in Malawi, for example, that there is a high prevalence of the wild type variants, PF81. In this case, there is a lot in seeing this kind of data. There is evidence that we don't know that much about all the contributing factors to drug resistance. Our suspicion here is that PFCRT and PA81 are in epistasis and maybe working together to lead to the phenotype that we are experiencing against chloroquine. Now, taking from the genomic data, we are improving on the things we're doing in the lab in vitro to understand how parasites evolve towards resistance. And we've developed an assay, for example, in the Gambia, that's called the Parasite Survival Rate Assay. And what we do with this assay is to expose parasites over 3 days to drugs, and we maintain the drug concentrations and determine how viable they are by looking at the rate at which they reinvade fresh erythrocytes. And we see, for example, here that this is a drug-free medium, and this is a medium that has lumefantrine. And you can see one parasite here continues to grow over 72 hours, despite exposure to more than 10 times the mean IC50 of lumefantrine in the Gambia. So this is evidence that using these methods, we might be able to identify isolates that might be evolving towards drug resistance. Once we've identified these isolates, we test them not only to the drugs that are currently available or the old drugs, we test them against experimental drugs or candidate drugs that are being developed, like those that have been made available to us by MMV. And once we test these drugs against these parasites, we will try to identify these new molecules, the new molecules that may be able to kill the parasites that are developing increased tolerance. So beyond these, we also sequence the parasites and compare the phenotypes and the genotypes in some form of genome-wide association studies. Now, what is emerging from this recent analysis of phenotypes and genotypes is that beyond the loci that are present on chromosome 7, we are seeing some new loci, for example, the locus on chromosome 10, that is coding for an antigen. Now, this antigen has been suspected before, has been shown before to have an association with halofantrine resistance. And we are seeing haplotypes around this antigen that are shared not only within the Gambia, but the haplotypes are haplotypes that are similar to haplotypes from Southeast Asia. And this, to me, hypothetically, might be indicating that some of the haplotypes that we are seeing in some isolates might have been imported when we got the importation of CRT haplotypes or DHFR haplotypes from Southeast Asia, and might also be contributing to parasite survival against drugs. Now, put together in a nutshell, we need to continue to do a lot more investigation across the continent, not just in single populations, for several reasons. There is a lot of heterogeneity of the parasite across the continent, especially in West Africa, and those in the East are becoming more homogeneous. And there is increased communication across the continent. What we've shown here using microsatellite is that there is a lot of sharing of parasites across the continent, and even more so across the eastern part of the continent, where we suspect drug resistance might have emerged first, might have been introduced first from Southeast Asia, and then these moved across the continent to West Africa. So we now have a new project which is looking at this, looking at more phenotypes and genotypes across the continent, across 3 countries: the Gambia, where there's a lot of low transmission, where we are sustaining pressure with drugs, and IRS as well. And there is Ghana, where there's moderate transmission, and in Nigeria, where transmission is fairly high, and there's also equally access to drugs. So we're in this new project funded by the EDCTP. We will continue to look at the evolution of the parasite towards drug resistance by doing more in vitro analysis, more genome analysis over time, as well as validating some of the markers that we've currently just found in the populations using forward genetic approaches. So this put together, it's very important work, driven not only by our desire to find out more about drug resistance, but also to train more young Africans in doing this kind of analysis. So my group in the Gambia has quite a number of young scientists and aspirants across the continent. The last time we checked, there were 6, but there are more than 6 countries now. We have loads of visiting staff funded by programs like Belgium, led by Djimdé, and the other funding sources that we got from across the globe, such as the MRC, which is directly funding me, the Malaria Gene, which is providing a lot of data to us, the Signal Center, where I am actually an international fellow, and the PDNA, of course, where we have lots of collaboration and networking across the continent, Work Big and Belgium, that are programs funded by the Deltas that fund the students that are doing most of the work that we have in the lab. We equally have collaborators in several institutions across the continent. For example, ERSF, Professor Umberto is my supervisor here at MRC, and he closely works together with me and advise me on some of the projects that I'm doing. I have a team of biostatisticians and informaticians working with the core informatics team that are also helping us to do the analysis that I've presented to you today. I hope this, in a nutshell, was what I had planned to present to you within 15 minutes on the work that we are doing both in the Gambia and across the continent. I hope this has been helpful. Thank you very much.
Thank you very much, Alfred, for this brilliant presentation and rich in information as usual. We will take questions at the end of the 2 presentations. So we'll move on to the next presenter, Nathalie Amvongo Adjia is from the Center for Research in Infectious Disease and the Institute for Medical Research and Medicinal Plant Study. Both the institutions are in Cameroon. Nathalie read animal biology and physiology at the University of Yaoundé and specialized in parasitology during her postgraduate studies. She has over 10 years experience working in vector-borne disease epidemiology and control. Her current research focuses on the bionomics and molecular basis of insecticide resistance genes in malaria vectors from wetlands across the volcanic chain of Cameroon. She works as a research officer at the Institute of Medical Research and Medicinal Plant Studies and a postdoctoral research assistant at the Center for Research in Infectious Disease, Cameroon, wherein she is mentored by experts in the field on the importance of applying novel genomic techniques to tackle insecticide resistance in Anopheles vector. Amvongo has been invited to report for the MESA correspondence program at PAMCA in 2019, at the PIMALPAR in 2020, and at the SDMH at 2020 conferences. She is a high achiever who is committed to pursuing a career in malaria genomics for vector control, and I'm sure we'll learn much more from her presentation that will follow. Nathalie, you have the floor.
Hello, everyone. During this 4th MESA webinar, I choose to present part of my research activities during my PhD studies. This work was entitled Exploring the Spread of Metabolic and Target Site Insecticide Resistance Alert in Anopheles fenestus vector population across a major mountainous landscape, the Mount Cameroon. As we all know, malaria remains an important public health problem. During the past 2 decades, significant progress has been achieved to reduce the burden of malaria and eliminate the disease wherever possible. And this was effective through vector control of mosquito vectors as a critical part of the global strategy to manage mosquito-borne diseases. With insecticide-based tool being the most important component of this effort. However, the widespread development of insecticide resistance in malaria vectors had negatively impacted the fight against mosquitoes in many areas. In Cameroon, malaria is the leading cause of morbidity and mortality. The major vectors are species of the Anopheles Gambia complex and the Anopheles fenestus group. The disease follows an equal distribution around the country, with high prevalence observed in wetlands around the Cameroon highlands. Insecticide resistance profile had been previously explored in Anopheles fenestus population from the Sudano-Sahelian zone and the forested area. In fact, 2 mutations, the L119F of the glutathione S transferase conferring resistance to DDT and pyrethroid, and the A296F of the GABA receptor gene implicated in the resistance to dieldrin have been identified in Anopheles fenestus population. However, dispersal barriers such as mountains are known to have demographic, evolutionary, and ecosystem-wide consequences. The presence of barrier to gene flow have already been explored and suggested to explain the restriction in the spread of insecticide resistance alert in other parts of the African continent, especially the Rift Valley area, the Rift Valley main continental-wide area. The Mount Cameroon, with its highest peak at 4,095 meters above the sea level, is the highest mountain in the West Central African zone. The zone is an area of great importance in the Cameroon agricultural sector, with industrial plantations of banana, rubber, and palm, activities that rely intensively to the use of agrochemicals, likely to select resistance in local mosquito populations. In addition, the Mount Cameroon constitutes a major landscape in the region with the potential to impact pattern of gene flow between vector population, but its potential influence on the spread of resistance alert remain unknown. Our study aimed at investigating the impact of the Mount Cameroon on the spread of GST2 and RDL resistance gene by studying the genetic diversity of both markers in Anopheles fenestus population across the Mount Cameroon altitudinal transect. Between 2010 and 2014, several entomological surveys were conducted in the Mount Cameroon region. We collected samples from the sea level at the lowland Tico, right up to 800 meters above the sea level at the local localities. Some mild altitude areas were also investigated and include the Mutengene area situated at 220 meters above the sea level and the Mianja situated eastward to the altitudinal transect at 305 meters above the sea level. Female mosquitoes were morphologically identified, and those of the Anopheles genus underwent further molecular identification. GSC2 and RDL resistance marker were genotyped in Anopheles finensis sample. In addition, genetic variability pattern based on the full-length gene sequencing were analyzed for both markers. In total, we collected 4,911 mosquitoes during the entire entomological survey. These mosquitoes were distributed among 4 genera, including Aedes, Culex, Mansonia, and the Anopheles genus, which was the most abundant. Morphological and molecular identification were conducted within female of the genus Anopheles, and this revealed the presence of 8 species, with the species of the Anopheles Gambiae complex and the Anopheles finensis being the most predominant. Genotyping results of GST2 and RDL mutation showed high frequencies of both markers in Anopheles finensis population in all localities. These resistance mechanisms are common to a number of Anopheline species, reflecting, in one hand, on the heavy use of DDT and pyrethroid for malaria control over several decades. In the other hand, despite the fact that cyclodiene are no longer used for malaria control in malaria control program, observation made with RDL in this study raised concern about the use of agrochemical targeting the GABA receptor in the agricultural environment, such as those found in the Mount Cameroon domain. It was interesting to note that land elevation seems to contrarily influence the frequency distribution of both mutations, with RDL resistance increasing with altitudinal increment, while with RDL with GST2 resistance increasing with altitude increment, while GST2 resistant specimens were more adapted to lowland area. Land elevation had previously been reported as an important predictor of the increase in pyrethroid resistance in another vector species, Anopheles gambiae, in West Africa, but these observations have not been made in East Africa. As such, this study highlights some uncertainties of the potential influence of altitude on the maintenance of insecticide resistance in malaria vectors under some specific environmental conditions similar to that of the study area. More specifically, GST2 homozygote resistance dominated throughout the study area, whereas heterozygote resistance for the RDL marker were the most predominant at high altitude, but this genotype was absent at the low altitudinal level. Therefore, understanding the factor which could possibly explain the persistence of RDL-based resistance mechanism in Anopheles tunestus are greatly needed in order to get insight on the resistance management. The presence of DST2 and RDL-based resistance was further confirmed after sequencing by the detection of a nucleotide change at the codon 119 for DST2 DNA fragment, inducing a nucleotide change of lysine to phenylalanine, while for the RDL, the characteristic change of a nucleotide at codon 296 was observed. This induced a change of an aniline to a serine. Genetic diversity pattern within the DST2 gene revealed a total of 14 polymorphic sites, which led to the formation of 12 haplotypes within this gene. Pattern of genetic diversity shows that the lowland tico and the midline mutengene situated at the same situated southwest of the altitudinal within the altitudinal transept are more closer to Anopheles tunestus population of the Meanja Eastward locality and the highland Bikoko. The cause of this clustering could be the similar geographic position around the Mount Cameroon mountain, or this could be associated to the presence of the Mount Cameroon, the mountain itself, which affect the population genetic structure and the speed of spread of GST resistant allele between Anopheles tunestus population. Comparatively, with 16 haplotypes found within the full-length DNA fragment of Adjia, genetic variability pattern shows that the lowland tico was more genetically distant to midland and the highland. Still, it could be hypothesized that the presence of the mountain influences the contrast in the RDL genetic pattern of resistance between vector population, thus suggesting the presence of barrier to gene flow between Anopheles tunestus population. For both genes, a strong selection process was observed, as seen by the limited numbers of mutational steps between resistant allele carrier as compared to susceptible allele carriers, as shown here by the haplotype network, as well as the maximum likelihood trees. Broadly, this study highlights for the first time the presence of GST2-based metabolic resistance and dihydrin resistance in the malaria vector Anopheles tunestus across the Mount Cameroon domain. High frequencies of both markers were observed throughout the study area, although the speed of spread of both mechanisms seems to be influenced by the presence of the mountain barriers. Signature of positive selection on both resistance allele was identified. And ultimately, our study emphasizes the need of molecular studies throughout similar mountainous landscape in order to fully elucidate the role of environmental changes on the acquisition of insecticide resistance. I could not end this presentation without thanking my mentors and all the organizations who facilitated this work. I'm grateful to the MESA team for giving me the opportunity to further disseminate my research findings. Thank you.
Thank you very much, Nathalie, for this presentation. Sorry, there have been some technical issues, but I hope most people were able to hear most of what you've said. We are now starting the Q&A session. I already see a few questions for Alfred. Maybe Regina, you can open your microphone and ask your question.
My question is in general, actually, across both resistance to insecticides and to drugs, which is there a concerted African effort by multiple funders to collect this kind of information and share it? Because it's very helpful to have these wonderful labs doing this research, including yours, Dr. Djimdé. But really, you're going to need to have a picture of how it's working across Africa. What's the progress on that?
Okay, Alfred?
I thought I was going to leave that to Djimdé to answer. Well, I think there is significant progress, because I think PATH and PMI, they're supporting various programs and various research groups in networks to generate data for drug resistance that I'm aware of. The GATE is also supporting molecular surveillance programs across the continent. We are receiving some funding from the National Institute of Health Research in the UK to do molecular surveillance for malaria, which is drug resistance and insecticide resistance in the vector in West Africa. So there are multiple parallel programs, but besides that, I think the PDNA Djimdé would obviously testify to this. It's working to coordinate lots of different scientists together with the NMCPs to be able to generate and share these data. Djimdé, I guess if I'm not accurate on this, please, you might want to add.
Yeah, well, actually, that's right. There is a beginning of a concerted effort. We've specifically, but I think what we need is to bring together the different ways of measuring this resistance and its spread. Like you said, PMI initiative with the across the various countries is more in vivo studies with very little of anything else. And some of the initiatives by the Gates now are more focused on genetic work. So the ideal situation would be to have a concerted Pan-African work where you bring together the in vivo studies, the in vitro phenotyping, and the genetic studies together. That is obviously going to be very costly, and because of that, it has been difficult to raise funds to actually have a comprehensive and Pan-African look at the situation. Hopefully we'll get there some days. The next question was by Linda. And Linda, can you open your microphone and ask the question to Alfred? Or if not, I can read the question. Have the findings from the research of your network had any impact on government policy or public health in terms of, for example, advising?
Oh, Djimdé has gone offline. Hi, Sonia. Can anyone hear Djimdé, or is it me?
No, he's frozen.
Yes.
I can finish the question. It was posted by Linda Anaga, who apparently can't log in either. Have the findings from the research of your network had any impact on government policy or public health in terms of advising on if a switch to a different malaria drug could be helpful, or the diversity of vaccine candidates under development? What alternative strategies could be considered? I think is what she.
Thanks very much, Linda, for that question. And I see, yeah, it's so put together. The national malaria control programs do take advice from the WHO, and obviously we interact with them and we share data with them. We are not in a position to recommend any change of policy directly at the national malaria control programs. So some of the data that we generate following genomic surveillance of malaria, the parasite and the vector, actually, they use it because they use that in stratifying populations. For the Gambia, for example, they use it in stratifying populations. And our wish is that as we progress in this and generate more data, this would directly impact the decision they make on which drugs is used where, and which insecticide is used in which locality. Because for Gambia, for example, there's increased heterogeneity in the populations as transmission has reduced significantly, and we're having different pockets with different profiles of drug resistance, which I may not have presented today, but there is clear differences between the East and the West, both for the vector as well as for the parasite, which has to be taken into consideration by the National Malaria Control Program. From them, they said, yes, they will do. So we're hoping to see this happen in the near future.
Okay, so Alfred, can you ask your question to Nathalie?
Oh yes, Nathalie, I was asking. I know that in the southwestern part of Cameroon, I come from Cameroon, and it's good to hear you talk as well. There are a lot of agro industries, rubber plantations, bananas, and they use lots of insecticides for vector control. Could this be having an impact on the prevalence of those alleles that you're seeing and the differences across the slope?
Yes, actually it's true. There is a massive use of those agrochemicals, and as they are polluting the breeding site, they increase the selection of resistant alleles at the larval stage, which is now disseminated at the adult stage. But in addition to that, there's also recently a massive use of insecticide based control tools such as insecticide bed net, spray, residual spraying in the houses, which can also modulate the resistance profile and exacerbate the resistance in vectors.
Okay, so I actually have a question for Nathalie as well. You mentioned that the mountain has sort of played the role as a sort of barrier for a flow of resistance genes. Can you elaborate on whether this is a sort of physical barrier in the terms of having different resistance gene on either on two different sides of a mountain, or if it is a height barrier, you have from different altitude, you see different resistant genes.
Yes, actually, it affects the altitudinal distribution. The altitude affects the distribution of those insecticide resistance gene, but also the position of the site, depending of the site at the mountain, is the southeast or west part, the vector population seems also to differ. So that's why we really think that the mountains has a role, and this has already been seen in the Rift Valley area in the Kenya.
Okay.
How a mountain can affect the distribution of insecticide.
Okay, so we have a question, two questions for Alfred. Francis asking whether the use of hydroxychloroquine for COVID-19 management in Africa can impact on the emergence of the CRT mutation.
Well, I think in one of our projects, we do speculate that it could happen. In terms of the level of pressure from hydroxychloroquine, I think we haven't quantified that, whether there's been adequate pressure to change the frequency of alleles at specific or continent-wide population levels. My feeling is that probably not. But we do know people have been taking all sorts of anti-malaria concoctions from leaves and stuff that have anti-malaria ingredients that might, for COVID. And these definitely might have an influence, which we should be able to measure in a short while, and we have direct intentions to do that in some projects.
Okay, so the next question is from Haile, who is asking what are the implications of the specific parasite population in Ethiopia on the prospect of malaria elimination there?
Well, we hope malaria should be eliminated across all populations. We just want that the specificity of some populations should be taken care of in terms of fine-tuning the intensity and type of interventions. For the parasite in Ethiopia, we realized that they are highly divergent, in which case their substructure is different from those from the rest of Africa, and they're very monoclonal. Now, the population in Ethiopia has also Vivax, and they've been using a lot of chloroquine and other anti-Vivax drugs, and these might be having an effect on P. falciparum. We do see that in one of the recent analyses that we did, is that there is also the difference in the prevalence in some of the haplotypes at PSRA and other drug loci in Ethiopia compared to the rest of the continent. So this might impact the way the population responds to drugs, but it requires further evaluation, like the other cases I've discussed.
Okay, so I think we are only 2 minutes left. I'll just thank both presenters for your very nice presentations and return the floor to Regina for closing remarks.
Thank you. I know that resistance is a constant, given the pressure that we're putting on both the parasite and the mosquito. And what came out here, but also came out in the Malaria Refresh when we discussed resistance, because we brought together the mosquito people and the drug people, is that the concepts are the same. The pressure is the same, and the data on both are going to end up being really important as decision makers try to optimize the interventions in their countries. But it's based on good science, which these were wonderful examples. Thank you so much. Thank you for participating to everybody. And we hope to see you for the 5th webinar series in October, which will feature Ogbonna Nwankwo from the University of Calabar in Nigeria and Jennifer Angaha from Jepaigo in Kenya. And they will be discussing below the target innovative ways to accelerate increase in IPTP coverage. We will be sending out a follow-up email with the recorded presentations and the summary reports of today's presentations. Thank you so much, and see you in October. And thank you so much to the speakers for your time in preparing and dealing with our technical challenges. Thank you, everybody. Have a good day.
Bye-bye.
Wow.